Well, hello, AP Bio. Here's your lesson for today. We are going to finish the immune system. There's just a few picky objectives that really aren't that difficult. There's a pretty big graph that is a little more difficult, and then just tie up some of the other loose ends. Review a little bit from class on Monday and from last week, Friday. So, all right, if we take a look at the schedule, you can see we have the primary versus secondary assignment. Shouldn't take you very long. We have 11 or 12 slides here for notes. Some of those should be review. And then the antibody questions. The first half I thought kind of came from Friday's lecture. You might need to go back and look at that again. And the rest from today and Monday. So if you remember all that stuff, it shouldn't take you too long. If you need to go back and review it, I think that's good too. But by the end of today, you should have all of the immune system objectives completed and be ready for the next chapter, which is endocrine system, which is hormones and things. All right, so let's look at this first assignment here, primary versus secondary response graph. So if you look at this graph, you're going to have to read what's happening here. So this is how many antibodies are in the blood. So it's one way that we measure how active your immune system is. And this blue line is antibodies to A. So this would be the first disease that we're studying here. And then also notice this is a log scale. So 10 to the 0 is 1, 10 to the 1 is 10, 10 to the 2 is 100, 1,000, and then 10,000. So it does increase a little bit more dramatically with the log scale. And then we also have the primary exposure to antigen A. Please be careful with this graph. It is not on day 0. It is maybe on day 2. And then it takes a while for the immune system to recognize the antigen, find some helper T cells that match it, and then activate T and B cells. B cells to make the antibodies and T cells to make the cytotoxic T cells. So really the immune response doesn't even happen for another 4 or 5 days. So this is the period when you're sick. And maybe a few days here too. And then once the antibody production kicks up, then you don't get sick. On the back end here is when the memory cells are made. So you will have some memory cells made with B cells and some memory cells made with the T cells. And so then next time you get exposed to this disease, this antigen, they call that a secondary exposure. So here we are on day, I don't know, day 29 or 30. Will really kick up antibody production, and it's several orders of magnitude higher, 2 or 3 orders of magnitude higher, which and it's more rapid too. Look, it only takes a couple days to kick up this high. It takes 1 day to kick up to this high, which was the original response. And so it turned out the second time you don't get sick. So this is the reason you don't get seasonal cold, get better, and then get it again in a week or 2. You are immune to it. Okay. Your immune system has those memory cells. You don't get sick again. This is also a reason why vaccines work. So instead of giving you the actual disease, we would give you a weekend or even just like the protein markers from bacteria or virus. And so then your body will recognize that as an antigen and make antibodies and go through the clonal selection and then store that in the long-term memory. So that's really nice. We don't get things like measles and mumps, and you guys don't get chickenpox because you got the vaccine for that. And so the vaccine would give you this response. You still wouldn't get sick because it's a weakened form of the virus or a disabled form of it. And then if you actually got exposed to the real disease, this would kick in and you wouldn't get sick, or you'd have a much more mild thing. You might even not even know. like one drip of the nose or something, or a few coughs for a day. Might not even be that sick. So there's quite a few questions here. Try to get within a day or two, as close as you can. Similarly with these numbers on the y-axis, get as close as you can. All right. The second graph here, it's the same thing with antigen A, but then we have a second kind of disease. And so maybe if this one was chickenpox and then this one was measles, I don't know, but this is disease B. And so that would be called antigen B. So when you get exposed to this one, you're going to go through the same thing again. Here you get exposed to both A and B that day. You must have been in a crowded elevator with some sick people because you got exposed to both of them. And within a day, you have a large immune response to antigen A because you have those memory cells and you don't have to go through clonal selection and produce lots of new cells to make antibodies. You have that machinery ready. And then contrast that with exposure to antigen B here in red. This would be your primary response to this one because it's the first time you've been exposed to it. Okay. Now, with the seasonal colds, you might get one next year because the virus does mutate enough that these memory cells that you made from last year aren't effective the next year. Same thing with people that get COVID like 2 or 3 or 4 times. The antigen has changed enough that the body doesn't recognize it, and so this secondary response doesn't occur. So you'd have to get another vaccine, an updated vaccine, or power through it. So make sure you give accurate numbers here and know about primary and secondary immune response, and then differences between antigen A and B. That one shouldn't be too bad. And then turn that one in here. And then let's look at the notes for today. So here's kind of the same graph again. It gives a little bit more explanation here. Okay, function of antibodies. This one should be reviewed. There was from Monday, there was 3 different functions here. One is neutralize. They surround the pathogen, engulf it so it can't infect any cells or cause any damage. Opsonize. Okay, this is when it gets eaten, and then it will display that. It helps mark these for destruction. So the phagocyte is shown here, that large eating cell or that macrophage. And because these cells are marked by the antibodies in purple, then the phagocyte or the macrophage will eat them more easily. And so once they do that, then they are destroyed. And then also the macrophage can display that antigen to helper T cells. The third way that antibodies could work is they will match here to the complement proteins shown here. And then these proteins will be activated and inserted into the infected cell membrane. This is really cool. These proteins will just open up a big pore. And so the inside of the cell contents will leak out, and that infected cell will be destroyed. All right, active versus passive immunity. So active is what we've been talking about so far. It's when there's memory cells. It's when their antibodies are made by white blood cells. It's when there's cytotoxic T cells and macrophages actively going against the disease. And then contrast that with passive immunity. Passive immunity is when you would get antibodies from another source. So you don't have cells that are making the antibodies, but you could get them from somewhere else. There are 2 ways to get these antibodies. One is through the mother's milk when you were a baby. And so this is one of the reasons babies that are breastfed are a little bit more healthy their first year of life. Number 2, and you can actually have these antibodies produced in the lab. We will filter them out of the cells that are making them, and then you could inject them into a sick person. This is one treatment that they tried during COVID, and it worked to varying degrees, and it helped people not die. I mean, they didn't get cured, but were less sick, and they had lower mortality rates with that. But that's kind of a last hope deal to do. So that would be a passive immunity, injecting antibodies either through the milk or through a needle. And those antibodies are made outside of the body from someone else or in the lab. All right, allergies are kind of an exaggerated immune response. So you probably have a really good immune system if you have allergies, but it's really annoying too, because then you have a runny nose or watery eyes, or you're stuffed up and you're not actually sick. You're just exposed to pollen or other sorts of allergens. So what happens here is the antibodies are produced after the first exposure to a certain type of tree or dust or whatever you're allergic to, and attaches to these mast cells. Now it's going to look for these allergens, and whenever it happens, it will release histamines and overactivate your helper T cells, which will then make B cells and cytotoxic T cells, and really just activate your immune system to fight dust or dander or whatever you're allergic to. So that's kind of how that works. Antibodies are also used in the lab, and my chemistry teacher in high school actually helped develop this for I think it was 3M actually. So the antibodies are going to match to the hormone that women have when they're pregnant, and so this hormone will be increased, and it will help keep the pregnancy. It's called HCG, or human chorionic gonadotropin, and so this it is secreted then in the urine, and so all you have to do is pee on the stick, and if that hormone is there, it will be detected by antibodies that are in the stick. And if you're not pregnant, it doesn't work. So the way that this works is you have to wait like 4 or 5 days until after conception, because it takes a little while for the body to recognize that it's pregnant and for this hormone level to go up. So you might have some negative tests those first few days if you try it. And so you might want to wait 4, 5, 6, 7 days to get a more accurate reading on the pregnancy test. They will also you can do these tests at home, but when you go to the doctor, they will also confirm it with a lab test in your blood. We can detect this elevated level of hormones in your blood as well. So what happens is the antibodies, if they match, they will actually form the second line. So they will bind to the hormone, and the antibodies will form that line. And then there's a second line to show that the test is completed. So if the antibodies get all the way to that line, they will just bind to that line, and that will show that the test is completed. Autoimmune disease is also a problem. This is when the immune system will attack the body itself by mistake. Okay, and so that auto is for self, and the disease is when it really starts to hurt. Some of these diseases, arthritis, lupus, MS, there's quite a few other ones where the immune system will just attack certain parts of the body by mistake. And so how do we help people like that? So there's actually a way that we can help eliminate some of this. And the body does it naturally by apoptosis. It will find those lymphocytes that have antigen detectors for our own cells and destroy them. Remember, apoptosis is a pre-programmed cell death. So here's kind of how that works. We have a stem cell. It makes a bunch of types of helper T's and helper B's. If this would match with cellular markers in our own body, they would be destroyed normally. If you have an autoimmune disease, it won't be destroyed. And so then when those bump into our cells, they would attack our cells, which then you have different symptoms. So normally then, if we have an antigen from outside of the body, it would match with one of the cells. And usually there's thousands of these different types instead of just 3 or 5. But for simplicity, they just put a few here. And if it matches one, that would be the end of clonal selection. So that might find a few days for that antigen to get a match. And then once there's a match, this cell will go into rapid mitosis. It will copy and divide and divide and copy, divide, and make lots and lots of B cells or cytotoxic T cells. Some of those will stay around after the infection as memory cells, and most of them will turn into plasma cells and just crank out antibodies, hundreds, thousands of antibodies, millions of antibodies. And those will circulate in your blood. And when they bump into the pathogen that they now match, they will bind, disable, mark for destruction. And you'll get better in a few days. Now, exertion and stress also play a role in the immune system. Exercise seems to help your immune system. Stress seems to hurt it. Rest is also important for a good immunity. There's certain things in your diet as well. If you don't have enough vitamin C or other vitamins, it could cause a more weakened immune system. So this is kind of a summary. Here's a COVID virus, and here's the immune response to that. Let's see if I can zoom in on it. There we go. All right, so here's the virus. The RNA is inside of this capsule or envelope. There's a membrane outside of that, and then there's these protein markers on the outside. Okay, they're called spike proteins. And those spike proteins will bind with this ACE2, which is a receptor on human cells. And so one of the ways that we were treating COVID was blocking that receptor, these ACE2 blockers, which is pretty cool. And then the virus could never actually get into the cell. All right, so when there's not a blocker there, the spike protein will bind, it will open up the host cell, it will take over the machinery there and replicate hundreds, thousands, millions of virus particles. It will burst out of that host cell, destroying it, and go out to infect other ones. Now, luckily for us, we do have some macrophages that would eat this virus and display it. We can tell this is a macrophage because it has a class 2 MHC presenting molecule here. And so that is going to go to the helper T cell and activate it to start fighting this virus. And that helper T cell will then release some cytokines. It will bind to a B cell and say, hey, go find this one, make antibodies to kill it. And so that B cell will replicate. It will make hundreds, thousands, millions of plasma cells, and those will each make hundreds, thousands, millions of antibodies that will circulate throughout your body. They will attack, surround, disable, mark for destruction, these virus particles, and hopefully you get better. Some of these B cells then, after the infection, will turn into memory cells so that hopefully you don't get sick. So like when I went to Mall of America and I got my COVID vaccine, it would present, it kind of skipped this part of infection, and it would just show you that spike protein. And you show that to the cells and activate this immune pathway. I did make some antibodies after that vaccine, but the bigger important part, I got some memory cells. And so then if I got infected out in real life, hopefully I wouldn't get sick, or I would get a much more mild version. So here you can see a virus that has been completely surrounded by antibodies, and that would be called neutralization. Okay, it was neutralized, it was disabled, and then it gets marked for destruction. All right, so that is the notes for today. I like the summary at the end. Make sure you do the antibody questions as well. These first ones kind of talk about the structure. So go back to Monday's notes for that one. And then these second ones talk about the antibody function, which we talked about twice, and self-tolerance was for today. So hopefully that's 8 quick questions for you. You can get through that, get that one turned in. All right, well, have a good day, AP Bio, and we'll see you tomorrow.